Hello everyone. Today we are going to see about fermentation media. Fermentation is the process in which a substance breaks down into a simpler substance using organism. The media used for the growth of microorganism in industrial fermentation must contain all the elements in a suitable form for the synthesis of cellular substance as well as the metabolic products. There are two types of fermentation media used in industries. First one is the synthetic media and second one is the crude media. Now let's see about the synthetic media. Media with all the essential constituents in a pure form in the desired quantity represents synthetic media. Use of this type of media in fermentation is not visible, and synthetic media is useful in the field of research as each and every component is chemically known and the exact composition of nutrients is predetermined. In synthetic media, variation in levels and concentration of nutrients can be controlled. It is very useful in controlling the growth and yield of product in a lab environment. It is used to determine the metabolic pathway and used in the synthesis of products. Next, we will see about crude media. The non-synthetic media with naturally available source are better suited for fermentation. Crude media is generally used on an industrial scale for fermentation process. Crude media contains a rough composition of media required for fermentation. It gives high yield of product and contains undefined source of ingredients. Crude media contains high level of nutrients, vitamins, protein, growth factors, antiforming agents and precursors. It is important to ensure that crude media should not contain toxic substance that could affect the growth of bacteria and yield of the product. Next, we'll see about the ingredients of crude media. First one is the inorganic nutrients. Crude media contains inorganic salts containing cations and anions along with a carbon source. Sometimes fermentation microorganisms have a specific requirement of ions like magnesium ions, phosphates or sulfates. These requirements are fulfilled by addition of these ions to balance the crude media. Next, we will see about the carbon sources. Simple to complex carbohydrates can be added to a media as a source of carbon. Different sugars like mannitol, sorbitol, organic acids, fatty acids, proteins, peptides can be chosen as a source of carbon. The selection of carbon source depends upon the availability as well as the cost of raw material. In most of the fermentation media, crude source of carbon is added. Next, we'll see about simple carbohydrates and complex carbohydrates. Simple sugars or semi-purified polysaccharides and sugar alcohols are added. Source of simple carbohydrates are blackstrap molasses, corn molasses, beet molasses, sulfide-based liquor, hydrol, cannery waste. And complex carbohydrates, which is the source of starch, corn, rice, rye, millet, wheat, potatoes, etc. Source of starch cellulose are corn, cobs, straws, wood waste, sawmill, etc. Next, we will say about the carbon sources, that is the molasses, which is a byproduct of sugar industry and is one of the cheapest source of carbohydrates. Molasses are concentrated syrups or mother liquors recovered at any one of the several steps in sugar refining process, with different names depending on the step from which it is recovered. Blackstrap molasses from sugar cane is normally the cheapest and most used sugar source for industrial fermentation. Sugarcane molasses and sugar beet molasses are commonly used. Besides being rich in sugar, molasses also contain nitrogenous substance, vitamins, and trace elements. Next is malt extract. It is an aqueous extract of malted barley containing about 80% carbohydrates like glucose, fructose, sucrose, and maltose. Nitrogen compounds constitute around 4.5%. It is proteins, peptides, amino acids, purines, and pyrimidines. Malt extracts are useful carbon source for the cultivation of filamentous fungi, yeast, and actinomycetes. Next, we will see about starch, dextrin, and cellulose. The polysaccharides starch, dextrin, and cellulose are not as readily utilized as monosaccharides and disaccharides, but can be directly metabolized by amylase producing microbes, particularly filamentous fungi. Their extracellular enzymes hydrolyze the substrate to mixture of glucose, maltose, or maltotriose to produce a sugar spectrum similar to that found in many malt extracts. They are frequently used for the industrial production of alcohol. Due to its wide availability and low cost, the use of cellulose for alcohol production is extensively studied. Next is about the sulfite waste liquor. The sulfite waste liquor is derived from paper pulping industry after wood for paper manufacture is digested to cellulose pulp. Waste liquors from coniferous tree contain 2 to 3 sugar and which is a mixture of hexose and pentose. Next is whey. Whey is a byproduct of dairy industry and is produced worldwide. Most of it is consumed by humans and animals. Whey is a reasonably good source of carbon for the production of alcohol, single cell protein, vitamin B12, lactic acid, and gibberellic acid. Storage of whey is a limiting factor for its widespread use in fermentation industry. Methanol and ethanol. Some of the microorganisms are capable of utilizing methanol or ethanol as carbon source. Methanol is the cheapest substrate for fermentation. However, it can be utilized by only a few bacteria and yeast. Methanol is commonly used for production of single-cell protein. Ethanol is rather expensive. However, at present, it is used for production of acetic acid. Next, we will see about the nitrogen source. Salts of urea, ammonia, and nitrate can be used as nitrogen source. When fermentation organisms are non-proteolytic in nature, few forms of urea, ammonia, and nitrate are used as a source of nitrogen. When fermentation organisms are proteolytic in nature, animal and plant raw material is used, like distilleries, dry solubles, casein, cereal grains, peptone, yeast extract, hydrolyzed, and soybean meal. The nitrogen supplied to fermentation microorganism may come from inorganic or organic source. So first we will see about the inorganic nitrogen source. Ammonium salts and free ammonia are cheap inorganic nitrogen source, particularly in industrialized countries. Ammonia can also be used to adjust pH of the fermentation. However, not all the microorganisms are capable of utilizing them, hence their use is limited. Next is organic nitrogen source. Urea is fairly a good source of nitrogen. However, other cheaper organic forms of nitrogen source amino acids and proteins are preferred. Corn steep liquor. This is formed during starch production from corn. Corn steep liquor is rich in nitrogen and is very efficiently utilized by microorganisms. Concentrated extracts generally contains about 4% nitrogen, including several amino acids. Its first use in fermentation was for penicillin production in the 1940s. The extract composition of the liquor varies depending on the quality of the corn and processing conditions. Yeast extracts, they contain about 8% nitrogen and are rich in amino acids, peptides, and vitamins. Glucose formed from glycogen and tetrahallose during yeast extraction is a good carbon source. Yeast extracts are produced from baker's yeast through autolysis or through plasmolysis. Yeast extracts are very good source for many industrial microorganisms. Next is the soy meal. After extracting the soybean oil from the soybean seeds, the left out residue is soy meal. It is rich in proteins as well as carbohydrate content. Soy meal is often used in antibiotic production. Next one is peptones or protein hydrolysates. The protein hydrolysates are collectively referred to as peptones and they are a good source for many microorganisms. They are prepared by acids or enzyme hydrolysis of peptones include meat, soy meal, peanut seeds, cotton seeds, and sunflower seeds, casein, gelatin, keratin. The protein, namely casein, gelatin, and keratin, can also be hydrolyzed to yield peptones. In general, peptones derived from animal source have more nitrogen content while those from plant source have more carbohydrate content. Peptones are relatively more expensive, hence not widely used in industries. Growth factors. Crude media constituents provides enough amount of growth factors, so no extra addition of growth factors is required. If there is a lack of any kind of vitamin or nutrients, growth factors can be added to media. For example, yeast extract and beef extract. Next, we'll see about the growth sources of growth factors. So, growth factors are very expensive in pure form. Thus, crude sources are preferred. Yeast extract is rich source of almost all growth factors. Generally, the substrates derived from plant or animal source in crude form are rich in mineral content. Sometimes, however, mineral supplementation may be required. Thank you all for watching this video. Have a nice day.